Hey guys, and welcome to a quick bio video. I know this looks like quite of a weird format, but we're going to try and do our best to talk about how colored markers can help us think about our meiosis modeling. So a couple of things when we look at the overview. We have our two pairs of homologous chromosomes. So the way that we can sort of think about this is we have our first pair of homologous chromosomes. We have two copies of a chromosome 1 that is dad and mom, and then we have a second pair of chromosomes right here that are dad and mom. Maybe lining them up like this might be a better way of thinking about it. We have pair 1, pair 2, dad, dad, mom, mom. Now we have this in meiosis 1 because we have taken our two pairs of homologous chromosomes, 1, 2, 3, 4 chromosomes, and we've duplicated each of them. So we have two copies of each chromosome. Now on these chromosomes, as you'll see with your packets, there are loci, specific regions of a chromosome that contain a gene. And those genes are the following. We have hair color, we have eye color, hair type, we have dimples, we have freckles, and we have earlobes. So each of these locations on our markers, or our chromosomes, are a gene that codes for a trait. Do you have red hair or brown hair? Do you have freckles or no freckles? And you've inherited these traits based on who your parents are. So normally when we go to page 2, thinking about meiosis modeling in terms of gamete formation, the following is going to happen if we don't have anything unusual happen. During meiosis 1, we're going to be separating our homologous chromosomes. We're going to be using microtubules that attach to the centromeres here, so we've separated them. Next, during meiosis 2, we're going to separate our sister chromatids. Almost like mitosis. So what does that leave us at the end of the table? We have 4 different cells, 1, 2, 3, and 4, that contain 2 chromosomes, 1 copy each. This cell over here contains a paternal 1 and 2, a paternal 1 and 2, a maternal 1 and 2, and a maternal 1 and 2. So if I was thinking about what do I draw in the 4 circles down here, I would probably be drawing something very similar. And what that means is you have a 50% chance of inheriting or giving to your offspring all of the dad genes and all of the mom genes. That's what happens when meiosis creates gametes without anything unusual. So let's now talk about one of the unusual things that happen. To do that, I'm going to go back to my initial drawing, where I'm going to be putting my homologous chromosomes back in metaphase 1 of meiosis, meaning I have duplicated copies of 4 chromosomes that are 2 pairs, 1 and 2, that are about to be divided. And to do this, I'm going to look at the directions on the top of page 3. I'm going to roll a die, and I'm going to roll a die for each pair. If I roll in evens, the pair does not swap, and if I do roll an odd, it does swap. This is what we call independent assortment. During meiosis 1, the microtubules that bind to the centromeres, the kinetochores, do not have eyes. So while they can match the chromosomes with the appropriate pairs, they don't know which side each is on. I rolled a 2 for my first pair. This is not swapping. I rolled a 1 for my second pair. These are going to swap. I'm then going to undergo meiosis 1. It stayed right there. And then I'm going to undergo mitosis 2. So what does that mean? It means that I now have 4 cells that contain the following information. Parental 1, maternal 2, maternal 1, parental 2. I would be drawing this out in these 4 circles here, where I'd have 2 cells or two gametes with paternal 1, maternal 2, maternal 1, paternal 2. That means there's a 50% chance that my offspring inherit my mom's eyes, hair color, and hair type, but my dad's dimples, freckles, and earlobes. Alternatively, I have a 50% chance of inheriting my dad's eyes and hair color and hair type, and my mom's dimples, freckles, and earlobes. We're starting to create diversity. The general trend that you need to know is that when we talk about the possible outcomes of independent assortment with meiosis, we are going to take 2 to the power of how many homologous chromosome pairs there are in a cell. Humans have 2, 3, 23, I know I just did a 4, 23 pairs of homologous chromosomes. So the different outcomes that we could get if we lined up 23 pairs of blue and red markers like this is about 8.4 million different combinations. If you then multiply that by 8.4 million happening with your reproductive partner, we're talking about a huge amount of diversity, a lot of diversity. That should keep you pretty busy for independent assortment. The second thing we have to talk about is crossing over. And crossing over is going to be on pages 4 and 5. This is what crossing over looks like. During meiosis 1, the microtubules of the centrioles help line up our duplicated copies of homologous chromosomes. Occasionally, during S phase of mitosis and the cell cycle, there are tiny little breaks in the DNA that happen at identical locations of both homologous chromosomes. It's rare, but it sometimes happens. And this is because we are making huge amounts of genetic information. Sometimes we make mistakes. So if we look at these markers, there's tiny little breaks that we've made at the same location. When that happens, proteins called chaperones, which are sort of like chaperones, they help things happen, swap the DNA. So what did I just do? I swapped the gene, the allele, from my dad's eye color over to this gene that's mostly maternal. And alternatively, I took my mom's gene for eye color and I swapped it over with a paternal gene that's mostly dad. So when I now go and do meiosis 1 and 2, I'm going to get these results. I separated my homologous chromosomes. I then separated sister chromatids. I'm left with one cell. That's 2 parental chromosomes, 1 and 2. I have 2 almost parental chromosomes. Parental chromosome 2 still here. Parental 1 chromosome contains a gene for my mom. Alternatively here, mostly maternal, 1 paternal gene, completely maternal, and then 1 gamete that's all maternal. So I'll repeat this process. During meiosis 1, we can have the same amount of genetic information on homologous chromosomes that are lined up, swapping. And this is how we have what's called crossover events, or chiasmata, where we have 2 different chromosomes made up of recombined DNA. So how are you going to do this with your model? Well, if you look at the directions on page 4 at the top, you're going to do the following things. One thing to note is that it's more probable for your DNA to swap over in bigger sections than not. So if you remember the directions for how you set up your 2 homologous chromosomes, I believe that your first pair of chromosomes right here have 1 loci on the top and 2 loci on the top. So I would arrange my marker like this. The first loci is here. Here's my centromere, second and third gene. Alternatively, if you read the directions on page 2, it's the opposite. You have 2 alleles above the centromere and 1 allele below. 1, 2, centromere, 3. So I'm going to arrange my markers like thus. Let's see if we can get a good picture of that. And again, if markers don't work for you, there's going to be other ways we can think about this in class next week. So, what do my directions say? Let's think about how we can do this. So I'm going to read for this. First, roll for the single gene above or below the centromere. I'm going to roll that, a 4. If you roll a 2 through a 6, it will not swap. So I just rolled for my homologous chromosome pair 1, where the 1 is above. I did not have a swap. I'm going to roll now for the pair below. I rolled a 2. If you roll a 2 or a 4, the genes will swap. What that means is I'm taking my big section right here, which has maternal genes for eye color and hair type, and I'm going to swap it over with my parental gene. I did this because I rolled a 2, which means that the probability of having the larger section, the bottom section of my first chromosomes, it worked. I had a high enough probability that the 2 swapped. So now I'm going to go to my chromosomes number 2, where I have the same thing except they're oriented in the opposite direction. So this would be chromosome 4, 5, 6, not chromosome, alleles 4, 5, 6 on my dad and mom's side. I'm going to roll the first die for the single gene here, which are the caps of the markers. I rolled a 6, doesn't swap. So in this game, the probability of getting a single allele to swap is pretty low. I'm going to roll again, and I rolled a 4. So the genes will swap. So what that means is if I'm looking at my 2 markers, like this, the 2 in the middle are going to swap the larger section. I'm going to do that now. Here's what my results look like. I have 1, 2 chromosomes that are completely paternal. Then my parental chromosome still exists here, but it's only parental for the first allele. And the second 2, second and third, are maternal. Then I have 4 and 5 that are maternal, with the 6 being paternal. Then I have mom, dad, 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 mom, mom, mom. I've created a lot of recombinants. I'm going to put these 2 here, these 2 here, these 2 here, and these 2 here for meiosis 2. So let's go through these options. My first option had no recombinants. This is paternal alleles for genes 1 through 6. Eye color, hair type, dimples, freckles, and earlobes. Here I had 2 crossing over events. In my first outcome, the top first allele, which is the gene for hair color, did not change, but eye color and hair type swapped over. So these are dad, mom, mom. Same thing here for my second chromosome. 4 and 5 were swapped over for mom, and the 6 is parental. Same thing here, but opposite. First chromosome remained mom, but the second and third be swapped over for dad. And same here. 4 and 5 for dad, and then 6 for mom. And then finally, my last gamete is 100% maternal. So if I were to draw these out here in this circle, I would be drawing these outcomes right here in terms of my recombinants. You're going to want to do this 3 times. So I always think that it's important to sum up an idea before we move on. So I'm just going to go over everything one more time. That way, if you have lingering questions, you can maybe get a little bit more insight. If this is still confusing, don't fret. It's not going to be the first time you get it for everyone. This would be a good thing to come into Flex or during work time to make sure you understand. So when I look at my marker pairs here, I have 2 pairs, 1, 2 of homologous chromosomes, mom and dad. Sometimes independent assortment means homologous chromosomes can swap over, and when we do meiosis, that results in complete chromosomes combining to make unique versions of mom and dad genes. Alternatively, in rarer cases, we can have even more discrete micro levels of genetic recombination, where 2 homologous chromosomes next to each other can swap identical parts of genetic information. This happens less, but it still creates recombinants of different types of genetic information. Excuse me. I hope this video helps. The general gist of where we're going is we need to understand how and why each of you looks different when we compare yourself to your parents and your siblings, and how your overall fitness is dependent upon that recombination of genes that you inherited when you were fertilized, and what that means for your own children someday. Let me know if you have any questions. I'm here to help. Have a good weekend.